let's talk about one of the most incredible missions happening inside your body right this second. It's the monumental task of copying your entire genetic code. This isn't just any copy job. It has to be done perfectly, trillions of times over. It's literally the foundation of life as we know it. You know, it's pretty wild to think that every single one of us started out as just one cell, a zygote. That tiny cell held the complete DNA instruction manual, half from one parent, half from the other. And from that single starting point, you get trillions of cells that make up a whole person. The real kicker? Every one of those new cells, whether it's building your heart or your hair, needs a perfect, complete copy of that original blueprint. So that leads to the big question, right? How on earth does a cell pull off this incredible copy job? How does it take this massive, ridiculously complex instruction manual and duplicate it with near-perfect accuracy again and again and again? Well, that's the mission we're going to crack today. To really get a sense of the scale here, just look at this number. 3.3 billion. That's how many letters or base pairs are in the human genome. Trying to copy this is like, imagine retyping a phone book the size of 1,000 encyclopedias, billions of times, and only making a couple of typos in your entire life. The level of precision is it's mind-boggling. Now, before we knew exactly how this worked, scientists had a couple of big ideas. The first was called the conservative model. The thinking here was, maybe the original DNA helix just stays completely together, and a totally new one is built next to it from scratch. The other idea, the semi-conservative model, suggested that maybe the original DNA unwinds, and each of its two old strands acts as a template to build a new partner. And through some really clever experiments, it turned out the semi-conservative model was the winner. As you can see here, every new DNA molecule is actually a hybrid. It's got one old strand and one brand new strand. This is the secret sauce to its accuracy. By using the original as a direct template, the cell makes sure the copy is a truly faithful replica. But a job this huge isn't a one-molecule show. Oh no. To pull this off, the cell puts together this highly specialized crew, a whole team of enzymes that work together like a super efficient molecular assembly line. Let's go meet the key players. First up, we've got DNA helicase. Think of this enzyme as the safecracker. The DNA double helix is wound incredibly tight, held together by all these hydrogen bonds. Helicase's job is to get in there, pry that structure open, and just unzip the two strands so the rest of the crew can get in and read the code. And here you can see it in action. Helicase grabs onto the DNA and, using ATP, the cell's universal energy drink, it just motors down the strand, unwinding everything and creating this Y-shaped opening. That opening is called a replication fork, and it's the main worksite where all the copying magic is about to happen. All right, and now for the star of the show, DNA polymerase. This enzyme is the master forager, the head contractor of this whole operation. Its job is to slide along that template strand, read the sequence of letters, and grab the right building blocks, the nucleotides, to build a brand new, perfectly matching DNA strand. But here's the thing about polymerase. It's a little bit picky. It has one very strict rule. It can only build in one direction, what we call the 5' to 3' direction. That's just a fundamental law of how the chemistry works. And this one little rule, well, it creates a fascinating puzzle for this cell to solve. So, here's the problem. The two strands of DNA are anti-parallel. Think of it like a two-lane highway where the traffic goes in opposite directions. If DNA polymerase is a car that can only drive one way, how in the world does it copy both strands at the same time? One strand is easy, it's going the right way, but the other one, it's going the wrong way. The cell's solution to this is just brilliant. It doesn't try to fight it. For the strand that's going in the right direction, the leading strand, polymerase can just zoom along, making one long continuous copy. But for the other one, the lagging strand, it does this clever little loop-de-loop and copies the DNA in short, backward stitches. These little pieces are called Okazaki fragments. And there's another little wrinkle. DNA polymerase is great, but it can't start from scratch. It needs a little something to grab onto, a starting block. That's where our ignition specialist, DNA primase, comes in. For every one of those Okazaki fragments on the lagging strand, primase lays down a tiny little piece of RNA called a primer. This gives polymerase the foothold it needs to get started. Okay, so now we've got a strand made of all these separate DNA fragments. The next step is to remove those temporary RNA primers and fill the gaps with DNA. But that still leaves these tiny little nicks in the backbone. So the final member of our crew comes in, DNA ligase, the cleaner. Its whole job is to act like molecular glue, going along and sealing all those nicks to create one single, unified strand.
So if you zoom out and look at the whole picture, you can see the entire crew working together in perfect harmony. You've got helicase unzipping the front, one polymerase cruising on the leading strand, and this whole other complex looping out the lagging strand with primase and another polymerase. It's not just a collection of enzymes, it's a beautifully coordinated, highly efficient machine.
So the copy gets made, and it gets made fast. But is it any good? I mean, a fast copy that's full of mistakes is pretty useless, right? Well, it turns out the cell has an absolutely incredible security system to make sure the quality control is top-notch. The accuracy is almost hard to believe. Thanks to its built-in quality control, DNA polymerase makes only about one mistake for every billion bases it copies. That's like making maybe 3 or 4 typos across your entire genome each time a cell divides. And this is all thanks to a remarkable skill called proofreading.
What's so amazing is that polymerase is constantly checking its own work. As it adds a new nucleotide, it sort of feels for the right fit. If it accidentally puts in the wrong one, the shape is all wrong. It feels that, hits the brakes, and uses an internal function that's basically a molecular backspace key. It chews back, removes the wrong letter, and then tries again with the right one. It's incredible. But, there's always another problem. When you unzip the DNA double helix, it creates a new issue. Imagine you have a rope that's tied down at both ends. If you try to pull the strands apart in the middle, the ends get wound up tighter and tighter, right? The exact same thing happens to DNA. As helicase powers forward, it creates this intense torsional stress that could actually stop the whole process cold. So to solve this winding problem, the cell calls in another specialist, DNA topoisomerase. This amazing enzyme works just ahead of the replication fork. It makes a tiny temporary break in the DNA backbone, allows the whole thing to spin and relax all that tension, and then perfectly seals the break again. Without this guy, our DNA would get tangled into an impossible knot. So this whole process is nearly perfect, but there's one final unavoidable flaw in the system. It's a consequence that plays out over a whole lifetime, and it's all about the endgame. The problem is right at the very tips of our chromosomes. On that lagging strand, once you remove the very last RNA primer at the very end, there's just no way for DNA polymerase to fill in that little gap. So what happens? With every single round of DNA replication, a tiny piece of the end of the chromosome is lost. To protect our important genes from being lost, our chromosomes have these long, repetitive buffer zones on the ends called telomeres. Think of them like the plastic tips on your shoelaces. And with every division, those protective telomeres get just a little bit shorter, and shorter, and shorter. This process isn't just a random quirk, it's a cellular clock. It's literally how our cells count how many times they've divided. Eventually, when the telomeres get too short, the cell gets a signal that it's getting old and it's time to stop dividing, a process that is deeply, deeply tied to aging. And that leaves us with a really profound thought. This entire process of copying our blueprint is a true double-edged sword. The super rare mistakes that do get through are what create the genetic variation that drives evolution itself. But they're also the root cause of diseases like cancer. And we know telomere shortening is a huge part of aging. So it makes you wonder, what if we could actually control this fundamental process? What if we could fix every error or stop our telomeres from ever getting shorter? What would that really mean for health, for disease, and for the very nature of life itself?